moving to the granular part of it. So as we know, fibrillar component has DNA and RNA, which is necessary for transcription process. In granular component, it is having rRNA, means ribosomal RNA molecules plus proteins, which forms ribosomal subunits. When we will discuss the process of transcription, we will see how ribosomal subunits are made from these rRNA molecules and proteins. So, ribosomal subunits. Size correlated with level of activity. Cells having high rate of protein synthesis means they require 20 to 25% of nucleus. The cells which are undergoing high rate of protein synthesis has many ribosomes, so they acquire 20 to 25% of nucleus space. Now, during cell division, condensation of chromatin into compact chromosomes takes place. Means when cell is undergoing cell division, then what happens? Condensation of chromatin. These thread-like structures which are present inside the nucleus, they undergo compact condensation. So, condensation of chromatin into compact chromosomes. Now, shrinkage and disappearance of nucleus takes place during this process. During this process, shrinkage and disappearance of nucleus takes place. After mitosis, chromatin uncoils and rRNA synthesis resumes. Now, tiny nuclei become visible and they fuse together to become large nucleolus. Now we will see chromatin, which are the molecular structures of chromosomes. We will now learn about chromatin. So by word you can understand it is the chromosomal part, the hereditary part of nucleus. So eukaryotic chromosomes have two broad components. One is nucleic acids and second is proteins. So in chromatin we will see that particularly in eukaryotic chromosomes there are two components. One is nucleic acids and the second is proteins. Nucleic acids mean it contains DNA and small amount of RNA. Proteins are in the form of histones, which are basic in nature, and we all know DNA is little bit acidic in nature. So the core histone proteins that we will see in chromatin are H2A, H2B, H3 and H4, and a linker histone which is H1. Here you can see nuclear envelope is there. Nucleus site of DNA replication. Chromatin is present in thread-like form. When we expand the nucleolus, here you can see DNA is present in the form of ribosomal DNA, which get converted into rRNA, and rRNA, basically the ribosomal RNA, is responsible to make proteins through the process of translation. Now when we expand a chromosome, the chromatin material, then here you can see the thread. When we expand the thread, here you can see the chromatin fiber like structure, and these are the histone proteins which are basic in nature, which are required for DNA to assemble and make a bead on a string like structure, which is necessary to bind the DNA and give it a compact structure. So histones are binding to the DNA thread, to the chromatin fibers, and giving us a nucleolus. So here with histone proteins, there are other proteins which are non-histone proteins, which are also required. And when it gets bind like this way, and then it forms a DNA double helix structure of DNA. So histones bind to the negatively charged DNA and gives it stability. So the function of histones is that it provide bind to negatively charged DNA, which provides stability to DNA. Now mixture of DNA and proteins give basic structural unit of chromosome, which is called as chromatin fiber. So chromatin fiber is what? It is mixture of DNA and proteins, and it provides basic structural unit of chromosomes. When this phase is made, then it is known as nucleosome after the whole binding. Here you can see a more expanded version where core of 8 histone molecules make a single nucleosome, and it is the whole thread are linked by linker DNA, linker histone protein. Histone H1 here you can see is binding a single nucleosome with the fiber. So when we study about nucleosome, what we see that it is simply a packing structure of DNA, and 146 base pair of DNA are wrapped around one histone octamer, because 8 are there, so octamer. So with histone octamer, when these are bound, then they form a single nucleosome. So basically octamer has 2 copies of 4 core histones. DNA length varies between species. Core DNA is DNA associated with histone octamer, and linker DNA is DNA between histone octamer 8 to 114 base pairs, 8 to 11 base pairs. Chromatin can be differentiated into 2 regions. So the chromatin that we see here, this whole part, this chromatin part is here in this diagram. You can see when this is the whole chromatin part in the nucleus. So the dense and dark color is euchromatin. Dark colored one is euchromatin between in the middle, and the lighter one, heterochromatin, is the peripheral part. So chromatin is differentiated into 2 regions during interphase and early prophase only. These are seen only in the interphase state, which is the longest stage, and early prophase stage. So what we see, euchromatin is lightly staining and heterochromatin is densely staining. So when we study about chromatin, so it is of two types, euchromatin, the true chromatin, and heterochromatin. So the middle one, which is lightly staining, is euchromatin because it is not present in a compact state, and heterochromatin on the peripheral side is densely staining. When we see the differences of both, then we see that euchromatin is lightly staining region and heterochromatin is darkly, because euchromatin is tightly packed chromatin fibers, therefore non-condensed, not visible with light microscope, undergo regular changes in morphology with cell division. It is the genetically active region, and heterochromatin is genetically inactive region. Now this replicates earlier during S phase. Euchromatin replicates earlier during S phase, while this replicates later during S phase. This is GC nucleotides rich and it is AT nucleotides rich. So we understood that it is staining very lightly and it is staining darkly. So this was it for nucleus part. Next we will further go with another organelle. Thank you so much for watching.